So now we will start talking about the translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor.
So keep in mind, translation needs different factors. Can anyone tell me what are the factors that needed for translation? The one they mentioned now? Can anyone tell me? No one. Yes.
The release factor or
Release factor.
tRNA.
Yes.
tRNA.
Yes.
في كمان.
tRNA.
ايش هذا حكى؟
Can you help him, Rand? Thank you, Sami. What else, Rand?
Okay.
أول mRNA
Yeah.
والribosomes large and small ما بعرف شو اسمهم بس large and small.
Okay, let's see it again. Let's see it again.
Translation is the synthesis of a protein from an mRNA template.
So it's a synthesis of protein from an mRNA template. The mRNA that is transcribed from the DNA, okay, this will be translated to make a protein, okay? So it involves several key molecules. These are the
This
key
process
molecules.
involves several key molecules, including mRNA, the small and large subunits of the ribosome.
We said that ribosomes are the factories of proteins. These are organelles that located inside the cell, which are involved in protein synthesis. Okay, so ribosomes, factories of proteins, also the mRNA and
tRNA.
tRNA, the one which will transfer the amino acid. Okay, so they could be bound together and form the protein.
And finally, the release factor.
The release factor. Okay.
The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and...
Now, there is something I would like to explain here. Now, when you write words, what do you use? We use letters, okay, to write a word. Tamam? Now, and word needs, some of them needs 2 letters, some need 3, some need 10, all right? But this is for the word, your language. But in the language of the gene, I don't call this language a word, it's called a codon. Okay? So this message is written by codons. 13 codons, red, orange, different colors. Now this codon is a language or a word in gene language. This is a word in gene language. In our language, English language, each word have different numbers of letters. في قلنا some of them have 2, 10, 3, 5, but in the gene word عندهم الكودون, we have for each codon 3 letters. Each codon, to give you a code, you need 3 letters. So هدول letters, they come from the bases. A, C, G, and U, which are on the RNA. We don't have T here. A, C, G, U. So you keep on mind, word in our language is equal to codons on the gene language. The codon as a word. We say that mRNA have a message. This message is coded in a code. Each code contains of 3 letters. All the codes have 3 letters. Not like in our language. They have all the same number of letters. Okay? Let's continue. So these codons are part of the RNA that have the message.
And moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA.
So in tRNA, they have codons, also the bases. They are opposite to the one here. يعني mRNA have A, U, G, so the tRNA will have the opposite. U with A with U, and C with G. يعني the complementary for this one.
RNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits. The ribosome moves along the mRNA.
Okay, now, what we need here, what we need to make here, or to bind here to make the protein, what are these parts that are binding? Met, and this orange thing. Who knows? What are these?
Amino acids.
Excellent. These are the amino acids. So, the amino acids, these are binded here according to the codon. The tRNA has a codon. We said each codon has 3 letters. Each codon translates an amino acid. I will show you here now another page in the book to understand more, but let's continue.
RNA, and the next tRNA enters.
So, tRNA will transfer, according to the codon they have, the amino acid needed to build a protein, and they will start to make the chain of polypeptide to make the protein.
These are the basic steps of elongation.
And here is elongation happen. Amino acid bonded to another amino acid to another, and elongation. They will be long chain, elongated. Can you see?
As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached,
so this is the ribosome, tRNA, and here is the mRNA, okay? Where they will move until they finish the whole gene. They going through each codon, and this codon will translate it to joining amino acid. After they come to the end of the codon, a releasing factor will detach this protein, and the ribosome will leave the mRNA.
the ribosome dissociates and the newly formed protein is released.
This is the protein. Now I will show you something here also. Just 1 minute, please. Right, just a minute, guys. Okay, I want to show you this page in the book. Okay, this will be very useful to understand more. All right, just 1 minute. Okay. And can you see it? Can you see it?
Yes, we can.
All right. Okay. So you have to keep in mind, the language of gene, okay, is made of codon. Our language is made of words. Okay. In our language, the word made of many letters or different numbers of letters. But in the gene language, each codon made of 3 letters. هلا each طبعا these 3 letters هم the bases. The bases اللي هم AGUC. Yes. If you look at this table here, you will see these 3 codons written, and each 3 codons have a name next to them. What that means? هلا each codon, which made of 3 letters, is coded for a type of amino acid. If you look here, UUU, okay, what that means? It means phenylalanine. This is a type of amino acid. So when this code is translated on the tRNA will know that I need to make transfer this type of amino acid, which is phenylalanine, from the cytoplasm connected to the chain. Now, if I told you that the genetic code matches a mRNA codon with amino acid, and each codon on mRNA, it will be match amino acid formation. Now, suppose you want to determine which amino acid is encoded for the CAU codon. Okay, on mRNA, we have codon CAU. Which amino acid is encoded to be made or to be bind to the chain? Let's see here, CAU. Where is the CAU? This is CAU. Hi, can you see it? And this one is histidine amino acid. Okay, so amino acids built in this way. Each codon has 3 letters. Each codon will code for an amino acid. And the building of amino acids in a polypeptide chain will happen in this way. Okay, we will repeat this again yesterday. Any question, guys? Any question? Okay, today I will send for you this video during the class, all right? And tomorrow, inshallah, we will repeat
Yes.
again. Yes. You have any-
خدت الاتندنس؟
Yes, 1 minute. Yes, now I finished the attendance. See you tomorrow, guys. All right? Bye.
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 Bye.